the male, the testis. However, in the case of the female, sometimes they talk of by default, you then have the development of the ovary. Let's talk about the gonad first, which is the ovary. Now, the germ cells come from the same source, that is the yolk sac. The other cells within the ovary are also originating from the sex codes or germinal codes that are invaginations of the coelomic epithelia. Once the germ cells reach the gonadal ridge, they proliferate, just like what we said yesterday, part of the proliferation stage. They proliferate, undergo a lot of mitosis to form large clusters of germ cells. These large clusters of germ cells we refer to as the oospheres. Oospheres. And that at that stage, that ovary would almost be called an oocyte at that stage of development. Now, the sex codes or germinal codes, which are from the coelomic epithelia, come to now surround this individual oospheres or the germ cells. They come to surround the germ cells. And the other tissue within that gonadal ridge, the mesenchyme tissue, will differentiate to form the supportive framework. I mentioned yesterday that the germ cells move in the entire area of the gonadal ridge, and as the proliferation continue on, this gonadal ridge is sort of bulging into the lumen and is attached to the posterior abdominal wall by the mesentery, the gonadal mesentery. That gonadal mesentery is eventually going to form the ligaments that attach the ovary and part of the uterus and the oviduct to the posterior abdominal wall. That is the broad ligament, the ovarian ligaments. These are part of the gonadal mesentery. As the ovary enlarges and as it sort of detaches from the posterior abdominal wall. Remember, during this enlargement, we have the oosphere and also the germ cells occupying the entire part of the ovary, what was the future ovary. However, we talked about those ones within the medulla degenerate. They degenerate. Only the cortical part, the one in the outer part, which is the cortex, are the ones which develop. So those within the medulla degenerate, and what we have in the medulla are blood vessels and some connective tissue. Blood vessels and connective tissue. In the cortex, we now have the germ cells and these codes, sex codes that have come to surround these germ cells. These sex code cells then differentiate to form flattened cells that we refer to as the follicular cells. Follicular cells. So we're going to look at the development of these follicles and also the follicular cells and also development of the oocyte within the ovary in the cortical part of the ovary, because the ones in the medulla degenerate. And then after that, we'll be looking at the derivatives of the Mullerian duct. We talked about the two ducts, the mesonephric duct and the Mullerian duct. In the case of the female, because the Sertoli cells do not develop, we do not have production of the anti-Mullerian hormone. And therefore, the Mullerian duct develop in the absence of anti-Mullerian hormone. So the Mullerian duct develops to form the oviduct, the uterus, and upper part of the vagina, like we had mentioned yesterday. So that's what you're going to look at and what can go wrong during this development. After that, we're going to look at the histology of these particular organs. We're looking at the histology of the ovary, histology of the oviduct, histology of the uterus, the cervix, vagina, and also some of the accessory reproductive organs. First, I'd like to invite any questions from what we discussed yesterday. If there was anybody that had questions, things that were not clear, I'd like to address that shortly. Any questions, put them in the chat so that we can be able to address them. Fine. Now, before we continue, let's just recap on part of the gross structures within the ovary. I mean, within the female reproductive system. What are these gross structures of the female reproductive system? And how are they related? The uterus, they say, is anteverted and anteflexed. Anteverted and anteflexed, except during pregnancy. Anteverted, you're talking about its relation to from the vaginal part. How is it related? How is it bent? And then anteflexed, how is it bent from the cervix, the entry of the cervix? How is it bent? So that's what they call anteflexion. The parts of the uterus, we have the fundus, the body, the isthmus, and the cervix. The fundus, just like what we talked about in the case of the stomach, is that large expanded part. And we are going to see that within the fundus, we can have sometimes malformations in terms of the arcuate type of uterus. Then we have the body, the isthmus, and the cervix. In terms of relations, the attachments, we have a lot of ligaments that attach the uterus and the oviduct and also the ovary to the posterior abdominal wall. And these ones are arising from the detachment of the Mullerian duct from the wall. As it continues to expand, it sort of detaches from the wall, but retains its attachment in the form of a mesentery. And that we refer to now as those mesentery form the ligaments. We have the broad ligament, teres ligament. In the parametrium, we have the transverse cervical ligament, uterosacral ligament attaching the uterus to the sacrum. We have pubovesical ligament attaching to the urinary bladder. And these ligaments have spaces between them. We have the paravesicular fossa, depression, parauterine fossa on either side of the uterus, and parasacral fossa. These ones are spaces in which you normally have peritoneal fluid, and this peritoneal fluid can accumulate. In a transverse section, so that we appreciate in the development, we have posteriorly the rectum, which we talked about in terms of the development of the cloaca, where you can have sometimes fistulas connecting the two because they have a common development, especially in the lower part of the reproductive system. And you can see the uterosacral ligament attaching the uterus to the sacrum so that it's maintained within the pelvic cavity. We have transverse cervical ligament attaching it on the transverse side, and then we have the round ligament. That round ligament actually extends to the labia majora. And then we have pubovesical ligament attaching to the urinary bladder. So it's very well secured. And remember, the urinary bladder and the uterus and part of the lower reproductive system originate from the cloaca, which we talked about yesterday. For the blood vessels, we have the uterine artery, we have the ovarian artery in the case of the ovaries, and this is from the abdominal aorta, the ovarian artery from abdominal aorta. Remember, this is developing from the gonadal ridge within, developing from the gonadal ridge, and as it develops from the gonadal ridge, the ovarian artery, also the testicular artery come all the way from the aorta. Then you have the uterine veins, which gross anatomy will be able to address. And then the lymphatic drainage, the fundus, the oviduct, the ovary, the upper part of the body. You need to know this drains to the lumbar region, and then the others drain to the external and internal iliac vessels. So why do we have this difference? Again, from the embryological development. From the embryological development, this is why we have the Mullerian duct, part of it from the Mullerian duct, part of it from the sinovaginal bulb, and also that's why we have this particular development, and gross anatomy will address the lymphatic drainage of that. For the female pelvic cavity, a transverse section just to look at the topography of the female pelvis. We've taken this from the specimens within the department, where we have the pubic part anteriorly. We have the fundus. This is what we call the fundus here, the rounded part of the bladder. This is the bladder, sorry, the bladder. And this is what we refer to as the fundus. Now, this way we talk about anteflexion and anteverted, anteflexion and anteverted. So if you take a section here, and if you draw an axis here, then you can see that the uterus is bent anteriorly. So that is anteversion. If you take a section through the cervix here, you can see that it is bent ventrally. So that is anteflexion. That will be addressed. But just to look at this diagram, and as we are going to describe the structure, you will note that it is covered by a peritoneum. It's covered by a peritoneum here, and then the broad ligaments that attach it, the posterior abdominal wall. This is a different section. They'll talk about pelvic diameters, that is for gross anatomy, which are very important during reproduction, during delivery. So you need to be very aware of these diameters during delivery, the diagonal conjugate diameter, the transverse diameters, and true conjugate diameters. Now, we have, in this position, you're talking about female external genitalia, where you have the labia majora and labia minora. And between that, the labia minora, we have the vestibule. The vestibule, in which that's the entry to the vaginal wall. And then the clitoris anteriorly. So we are going to look at this as the homologous structure of the penis. Just another section through the pelvis to demonstrate the structures, the female reproductive structures, and the ligaments that attach it, the posterior abdominal wall. Of importance, you would note here the oviduct. It is open to the abdominal cavity. This opening to the abdominal cavity is because it develops as an invagination of the coelomic epithelia, which we just described yesterday. So the Mullerian duct developed as an invagination of the coelomic epithelia. So for the development, we talked about proliferation of the gonadal ridge, within the the gonadal ridge is very close to the mesonephros, lateral to the mesonephros. And that is important to note that it's very closely associated with the kidney. And as we look at the kidney, we're going to see that part of the structures of the kidney, which we talked about yesterday, the pronephros, the primary part of the kidney, forming or being incorporated into the male reproductive system as the efferent ducts. We discussed this yesterday about the epithelial cords forming the epithelial cords in the male. So I'd like us to visit this and just ignore that this could be a male or a female, and we have the epithelia invaginating into the gonadal ridge as geminocords, or what we call sex cords, which we talked about yesterday. As this continue to expand, the whole of this will be attached. It will be a round structure, and the whole of this will be attached to the posterior abdominal wall by the gonadal mesentery, which eventually forms the broad ligament. The broad ligaments and ligaments of the ovary. Broad ligaments and ligaments of the ovary. Now, the germ cells, which have come from the yolk sac, proliferate, like I've said, and form clusters of germ cells. These clusters of germ cells are then invaded by the geminocords or sex cords and come to be surrounded by the sex cords. Those sex cord cells, I've said, differentiate to form the follicular cells, to differentiate the cells. I'd like you to know the there's detachment from the wall of the urogenital ridge. This detachment now is attached only by broad ligament, that eventually forms the broad ligament. We talked about the cloaca, and if you remember the cloaca, we have several parts of the cloaca. Initially, the cloaca, we have urorectal septum that divide the cloaca into the urogenital sinus and then anorectal sinus. Urogenital sinus and anorectal sinus. The urogenital sinus will have several parts, the upper part forming the bladder and the lower part forming the definitive urogenital sinus. Opening into this, we have both the reproductive parts, that is the derivative of the Mullerian duct, opening into that. But I'd like us to look at this also at this entry of the Mullerian duct. At the entry of the Mullerian duct, we have a development, a thickening of the epithelia in that area, and that thickening forms what we call sinovaginal bulb. Sinovaginal bulb, close to this entry, within the definitive urogenital sinus. That sinovaginal bulb grows towards and attaches to the Mullerian duct. At the area of attachment to the Mullerian duct, there's a thickening of the epithelia, and that thickened epithelia forms what you refer to as the hymen. It forms the hymen, which is a membranous structure, and this hymen will have some small perforations, or sometimes it might become imperforate. When it becomes imperforate, then that is a big problem because the menstrual flow will not be able to pass through. in the early stages. We also talked about this upper part, the phallic part, and that phallic part within the males develops to form part of the urethra, urethral fold, and urethral meatus, and then the fold. And then in the case of the females, that phallic part also develops to form homologous parts of the females, homologous to the males. For the female development, we normally have stages, what you call the induction stage, which is the initiation stage, and that is under the limb protein. Then invagination stage, that is of the oviduct, or what is eventually going to form the oviduct and the uterus. That's the Mullerian duct. And then elongation. That elongation continue on to the urogenital sinus, definitive urogenital sinus, to form the uterovaginal duct, and this under the Wnt genes and the Pax genes. So you can see at every stages, we have the genes involved in directing the growth of the cells. We normally have this Mullerian duct eventually towards the distal part coming very close together, forming a Y-shaped structure. The distal parts fuse. That part of the distal part which fuse of the Mullerian duct, as it close to the opening into the urogenital sinus, is what forms the uterus and the upper part of the vagina. So that fused part. When the two parts are fused, it forms a Y-shaped structure. But there's proliferation of the cells within that Y-shaped structure, so that we have a development of the fundus, and also we have the development of the common uterus and common vagina. That is at the area of fusion, there is degeneration of the cells that form the septa between that fused area. And that degeneration now leads into a common duct. Again, right from this, you can be able to distinguish an anomaly that can occur. One, that we might have one part of that Mullerian duct not being inducted and invaginating. So failure of that induction would mean you have only one part of the uterus. That's uterus unicornis. You can also have within that elongation and fusion at the distal part, that this fusion at the distal part can have the septa dividing these two after the fusion might fail to develop, or there might be incomplete fusion, so that you have two horns of the uterus, what you call uterus bicornis or the delphis. You might have the two completely separating and opening into the oviduct and developing separately. In which case, because the genes are still responsible for the development of these distal parts, those distal parts will be completely separate, where you will have two uterine horns, you will have two cervix, you will have two vaginas opening into the cloaca. So that again can persist, and we'll be looking at some of the malformations. So all those we refer to as Mullerian dysgenesis, as we'll be looking at later. Now, we also have like I said, at the entry of the Mullerian duct into the cloaca, the cells close to that proliferate and thicken. So that thickened proliferation grows towards the Mullerian duct, forming what we refer to as sinovaginal bulb. That part of the sinovaginal bulb forms the lower third of the vagina. the lower third of the vagina. This vaginal bulb is what forms the lower part of the vagina, about a quarter of the part of the vagina. So this is how the female tract develops. Now for the ovary, we've talked about the ovary in terms of development of the migration of the gonadal, migration of the germ cells, coming to be surrounded by the sex cord cells, which are the ovarian sex cord cells or germ cells, and then those cells differentiating to form the follicular cells, and then the other cells within the ovary in the intermediate mesoderm on the gonadal ridge forms the mesenchyme tissue that differentiate to form the supportive cells, the stroma, and other structures of the ovary. What can go wrong during this development? We have what we refer to as the streak ovaries. Remember, the ovaries are also developing under the influence of hormones, under the influence of, not hormones, sorry, under the influence of course hormones, and also under the genes. The genes direct the development of the ovary. So we can have malformations of the ovary. We have what we call streak ovaries. This is poorly developed ovaries, and then also what we call gonadal dysgenesis, where the ovary is like very poorly developed, underdeveloped ovaries. And again, this is because of the genes, two, because of lack of hormone direction. And this can occur during Turner syndrome, where you have only 45 chromosomes with one X chromosome, one lacking chromosome, or 46 with some extra chromosomes. So this can occur. We can also have accessory ovarian tissue. If you remember yesterday, I talked about the polyorchidism, where you have small testicular tissue apart from the main tissue. So similarly here within that gonadal ridge, the gonadal ridge might split at an early stage on one side. So if there's that split of the gonadal ridge, then each of those will develop also into some ovarian tissue. So that's what we have as accessory ovarian tissue. Most of the other malformations are related to the Mullerian duct in what we are calling Mullerian dysgenesis. So we might have a genesis of the Mullerian duct. That is where the Mullerian duct does not develop. And in this case, it can either be unilateral, that's on one side, or bilateral. And remember, the entire Mullerian duct developed to form the oviduct, the uterus, and the upper part of the vagina. So if it does not develop on one side, then you can have only one part of that developing. You will have uterus unicornis, only one whole part of the uterus developing on one side, either right or left. And then you might have failure of the oviduct to develop completely on one side or on both sides. If it is on both sides, then all the female reproductive tracts will not develop. Then uterus unicornis, only one Mullerian duct develop, the other one does not develop. So you'll have only one uterus on one side. Uterus didelphys, where you have failure of fusion. In failure of fusion, remember the Mullerian duct fused towards the distal part. So the proximal part of that, which is not fused, becomes the oviduct. So if it is failure, the distal part failed to fuse together, then you'll have separate uterus, separate cervix, separate upper part of the vagina, and even separate part of the entire vagina. All this open into a common part that will have been the cloaca. That is in uterus didelphys. Uterus biconnuate, where there is incomplete fusion of the upper part, incomplete fusion of the upper part that would have formed the uterus. So the uterus sort of splits into two horns, and then septate uterus, where there is incomplete degeneration of the septa that separate the fused Mullerian duct, septa that separate the two Mullerian ducts. So this will have completely two different cavities. So the uterus, instead of having a common cavity, will have two different cavities. And then an arcuate uterus, a small crest at the fundal part. And this is also very close to the, if this is partial fusion of the upper part, then you might have a depression in that part of the fundus, and that causes an arcuate part malformation. Sometimes we can have cervical weakness, and this cervical weakness can be because of reduced collagen synthesis. So any problem with collagen synthesis can lead to cervical weakness. Apart from this, we have androgen sensitivity syndrome, where we have a 46XY female phenotype. 46XY female phenotype. And this one here would have a functional testis. And here you are talking about the intersex situation, where you have both male and female reproductive organs. And we have what is called discontent identity. Discontent sexual identity is a central nervous system situation where people feel like they are females and they're actually they feel like males and they're females, or males might feel like they're females, but they feel and behave, but they are actually having genetically they are males, or genetically they are females. They have all the organs of male and organs of female separately. As a female, you'll have all the organs of the female, but there's that feeling that you are like a man. That's what you call discontent sexual identity. I think this is an area where there's been a lot of discussion, and people have said, okay, you find people dressing like females, behaving like females, whereas they are males, and you might have females also behaving the other way. So this is more of a central nervous system situation. Then we have the intersex situation, where we have ambiguous genitalia, what we call hermaphrodite. We can have what is true hermaphrodite and pseudo hermaphrodite. In pseudo hermaphrodites, you might have some you might have some organs. True hermaphrodite is where you have both male and female organs, You might have an ovary, you might have a testis. That's a true hermaphrodite, or ovotestis sometimes. So in this case, there was development of both, and this can be bilateral or unilateral. One side could be a testis, the other side could be an ovary, or both of them are on one side. Now, the germ cells in that particular case would be germ cells of the female, the oocyte, and also germ cells of the male. Then we have male pseudohermaphrodite, where you would have a testis, and then you can have an oviduct or the uterus because of some parts of the Mullerian ducts developing. So this is currently called intersex. The intersex, not the word hermaphrodite is not used as much now, but they use the word intersex. We have had cases where people get admitted to schools and they don't know they are in a girls' school and they have male organs, or they are female school and they have male organs, or in a boys' school and they have female organs. So where do you put them? These are some of the malformations that we'll have. In this case, uterus biconis, you can see the uterus have two different horns because of failure of fusion at the distal part, and then a septa also in the vaginal part. And then unicornis, where you have only one horn because failure of the Mullerian duct to develop. Failure of the Mullerian duct to develop on one side. And then this way we have a septate type of uterus, Biconis is where you have this didelphys, biconis where you have two horns. And here, usually we should be having a rounded part here, which is the fundus, but you might have a small depression. And when you have that small depression, that's an acute type of uterus. This is an MRI showing unicornis. There's only one horn of the uterus here. The other horn failed to develop because there was no Mullerian duct on that side developing. This is two horns, biconis. Well, this is the two horns. The other one is the didelphys. This is the uterus biconis. You can see the two horns here separating completely. And when you have that, you might also have it extending the vaginal part. And this is a septate type of uterus. Septate, there's a septa separating the two. This failure of complete degeneration of the area where the two Mullerian ducts fused together. In this septate also type, you might be able to see a depression here, which is an acute type of uterus. So this one is septate and also acute. Instead of having a rounded fundus, that's what we have as an acute. Let's now look at the ovary, the histology of the ovary. The ovary is a paired structure. Paired structure suspended within the pelvic cavity, by ligaments, the ovarian ligaments, the round ligament of the ovary, and suspensory ligament of the ovary. The round ligament of the ovary extends to the labia majora, and that is homologous to the ovarian. That's homologous to the gubernaculum testis. So that is also what we call gubernaculum ovary. And the gubernaculum ovary is also very important in the descent of the ovary. So it helps in the descent of the ovary so that the ovary finally comes to lie close to the pelvic cavity. The ovary is very important in storage and development of the oocyte. Storage and development of the oocyte. It's also very important in the production of hormones, specifically production of progesterone, estrogen and their precursors, estradiol. It's also important in the production of, sometimes they talk of production of prostaglandins. And this development is under the influence of the hormones produced from the pituitary gland, that is the follicle-stimulating hormone and the luteinizing hormone. Follicle-stimulating hormone and the luteinizing hormone, which are stimulated by the gonadotropin-releasing hormone from the hypothalamus. And remember, we talked about the short, the direct, and the indirect long loops, feedback loops, where the secretions from the ovary will influence the release of the gonadotropin-releasing hormone from the hypothalamus, but also it can influence directly the release of follicle-stimulating hormone and luteinizing hormone from the hypophysis. For the structure, the ovary is covered by a thin connective tissue capsule, which developed immediately below the coelomic epithelia, just like what we saw in the testis. This thin connective tissue capsule we refer to as the tunica albuginea. The tunica albuginea, the same as what we called in the case of the testis. This tunica albuginea is covered by a mesothelium, which is a peritoneal covering comprising of simple cuboidal epithelia. Simple cuboidal epithelia. That mesothelium is sometimes called germinal epithelia. It's germinal epithelia, but truly it's not responsible for the development of the germ cells, sometimes referred to as germinal epithelia. The ovary consists of a cortex, the outer part, in which we have developing follicles, and the medulla. And the medulla is where we have the blood vessels, large blood vessels, and also the connective tissue. Large blood vessels, nerves, and connective tissue enter at the medulla. So the medulla is the central part. And remember that it's only the germ cells in the cortical part that develop. The germ cells in the medullary part degenerate in the case of the ovary, which is opposite to that of the testis. The cells within the ovary are several cell types. We have the interstitial cells. These interstitial cells secrete estrogen in the prepubertal stage. And then we have the theca interna cells. These theca interna cells, the theca interna is a highly vascularized area close to the follicle, and is separated from the follicular cells by a membrane, which forms a basement membrane of the follicle. The theca interna area is an area which is a highly vascularized area consisting of slightly flattened cells. And these theca interna cells secrete estrogen. The theca externa cells are stromal cells. They are supportive cells. They consist of flattened fibroblast-like cells that are supportive in the ovary, that form the supportive tissue of the ovary. The other cell types that we have are the follicular cells. We'll be talking about those shortly. Follicular cells, they are derived from the germinal cord cells, cord cells, which are the ovarian sex cord cells from the coelomic epithelia. So those are the follicular cells, which I have said under the influence of follicle-stimulating hormone now form the granulosa cells. And we are going to look at those granulosa cells. The granulosa cells, after ovulation, under the influence of luteinizing hormone, now differentiate to form the lutein cells. Under the influence of luteinizing hormone, the granulosa cells form the lutein cells, and also the lutein cells are from the two sources. We have the granulosa lutein cells, and we have the theca lutein cells. Theca lutein cells are from the theca interna cells. Apart from this, the other ones are primordial cells. These are the primary oocyte, and then eventually the secondary oocyte, but primary oocytes. And these primary oocytes are surrounded by the primordial cells. So when you talk about the follicles, we'll have different stages of development of the follicles. These are ovary from the surface. we have a mesothelial covering made up of simple cuboidal epithelia. We have a very thin connective tissue immediately below that mesothelium that we refer to as the tunica albuginea. Tunica albuginea, made up of collagen fibers. Just like any other organ, this forms the capsule of the ovary. And from this capsule, we have the tunica, the supportive fibroblast cells entering into the ovary. You can see that blue staining is the connective tissue that enter into the ovary, giving it that supportive framework. Within the ovary, we have the follicles at different stages of development. Very close to the capsule, we have the primordial follicles. The primordial follicles, these are primordial follicles. Primordial follicles consist of the primary oocyte, and this primary oocyte is surrounded by flattened cells. This squamous cells, a single layer of squamous cells, is what we refer to as the follicular cell. The follicular cell rests on a basement membrane, separating it from the theca interna. That follicular cell, under the influence of follicle-stimulating hormone, then transform and become cuboidal. So we have a single layer of cuboidal cell. This is now what we refer to as the primary follicle. This we refer to as the primordial follicle. So the primary follicle consists of granulosa cells resting on a basement membrane, and this can consist of a single layer of granulosa cells, which are simple cuboidal, but the single cuboidal multiply to form several layers. This surrounds the oocyte, the primary oocyte. That primary oocyte is separated from the granulosa cells by a zona pellucida, a glycoprotein membrane that we refer to as the zona pellucida. As the number of granulosa cells increase, they multiply, there is a thickening of the wall, 2 layers, 3 layers, 4 layers, and as it thickens, now the oocyte, just like any other epithelial lining, will not receive adequate nutrients because this is an epithelia. Granulosa cells form an epithelia arrangement. The blood vessels are within the theca interna region. So nutrients must diffuse through the basement membrane, through the epithelia to reach the oocyte. We therefore then develop areas where you have fluid-filled cavities. These fluid-filled cavities are separations of the granulosa cells. As they separate, those single fluid-filled cavities coalesce to form a single cavity that we refer to as the andron. Before that, we'll be having several layers of granulosa cells, up to 8 layers of granulosa cells. In that case, we refer to that as a solid form of secondary follicle. Solid form of secondary follicle. That solid form of secondary follicle, we have spaces forming between the cells, separating. Those fluid-filled spaces is what we call the andron. So as the andron enlarges, this then we refer to as vesicular form of secondary follicle. Vesicular form of secondary follicle. You can see in this particular case, a small fluid-filled space. So that is the andron, a vesicular form of secondary follicle. But as this develops further, the lumen enlarges, the andron. This andron here enlarges. It enlarges so that we now have the several layers of granulosa cells several layers of granulosa cells, and there are those granulosa cells that come to surround the oocyte. So when the granulosa cells, like just what I'm drawing here, when the granulosa cells surround the oocyte, but do not completely form this type of arrangement, so that this part is filled with the granulosa cells, then we refer to that as a tertiary follicle. Tertiary follicle. But as the fluid increases in size, as the follicle increases in size, then the oocyte come to be surrounded by granulosa cells and attached to the outer layer of granulosa cells by a thin strand of granulosa cells. This thin strand of granulosa cells attaching the oocyte to the outer layer of granulosa cells we refer to as cumulus oophoros. Cumulus oophoros. And the granulosa cells that surround the oocyte, forming something like a crown, we refer to as corona radiata. Corona radiata. This is a crown sort of thing that surrounds the oocyte. Corona means crown. So corona radiata. Note here that there is a primary oocyte and is surrounded by this glycoprotein membrane that we refer to as the zona pellucida. Zona pellucida. However, the processes of the oocyte penetrate through zona pellucida and make contact with the inner layers of the corona radiata cells, the granulosa cells that are close. This is very important for cell-to-cell signaling. Cell-to-cell signaling. At this stage, when you have now the cumulus oophoros and the corona radiata and a very large lumen, we now refer to that as a mature follicle, also referred to as Graafian follicle. That is now when you have preparation for ovulation. But I would like us to look at this slide again. You will notice that the primordial follicle is very close to the capsule. So as this primordial follicle develops, it moves more internally, but retains its contact with the capsule. And this retention of the contact with the capsule forms what you refer to as an ovulation cone. An ovulation cone. This is where, once this follicle matures, this is where at the time of ovulation, there will be release of the ova through that part there into the abdominal cavity, that ovulation cone. So what happens after ovulation? Ovulation is under the control of, there's a luteinizing hormone surge at the time of ovulation. There's a rise in luteinizing hormone, sudden rise in luteinizing hormone, and this causes the sudden increase in the fluid in the androgen. So there's an increase in pressure within the androgen, and because the oocyte is attached to the granulosa cells by a very thin layer, then you have that being ejected out as ovulation. What then happens to that space? What happens to this space here after ovulation? When the oocyte, together with the corona radiata cells, are released, this androgen, the fluid-filled space, is now invaded by the granulosa cells here, and also the theca interna cells. Theca interna cells, because of the rupture of the blood vessels in the theca interna, and also theca interna cells invade this area of the andron. Together, because of the rupture of the theca internal cells, there is a mixture of blood and the granulosa cells. The blood and the granulosa cells form what we call corpus hemorrhagicum. A corpus hemorrhagicum. So a corpus hemorrhagicum, where we have granulosa cells suspended within blood vessels, within the blood clot. That blood clot gets resorbed. Once it gets resorbed, then those cells differentiate under the influence of luteinizing hormone to form lutein cells. It then forms what you call the corpus luteum immediately after ovulation. It forms a corpus luteum. However, unless there is implantation, if there is no fertilization, then the progesterone levels reduce. When those progesterone levels reduce, then you have the area where you have the lutein cells now degenerating to form a corpus albicans. However, if there is fertilization, then the trophoblasts, after fertilization, trophoblast cells produce human chorionic gonadotropin hormone. The human chorionic gonadotropin hormone stimulates the release of luteinizing hormone by the pituitary glands. So the luteinizing hormone then continue to maintain the lutein cells within the corpus luteum, so that you now have a corpus luteum of pregnancy. A corpus luteum of pregnancy. I think you should be very clear about that initially there is a corpus luteum, but it's not persistent. Then that corpus luteum will be maintained only if there is fertilization to form corpus luteum of pregnancy. If there's no fertilization, there'll be no trophoblast formed, there'll be no human chorionic gonadotropin hormone released. Therefore, there'll be no continuous release of luteinizing hormone. And therefore, the cells there degenerate and form something similar to scar tissue that we refer to as corpus albicans, a corpus albicans. So we need to be clear about that. Now, we are trying to compare here the development of the oocyte and the development of the follicle. Remember, the development of the follicles occurs at puberty. The follicles develop, but the oocyte development occurs at puberty. So we have the meiotic division, first meiotic division. At the prophase stage, there's an arrest of further development of the oocyte. So the follicles will have developed. So at birth, there'll be millions of follicles. Before birth, there'll be millions of follicles which are developing. But at birth, about 400,000, 400 follicles are developing at birth, as persistent at birth. The rest will have undergone degeneration. So before birth, we have mitosis, which we talked about as the germ cells undergo mitosis, the growth, then the growth phase, and then in the childhood stage, that's before puberty, to menopause, we then have the development of the oocyte. So at this stage, there's an arrest of further development of the oocyte. So the oocyte becomes inactive only up until puberty. That is when it develops. For the follicle, at the time of birth, we have a primordial follicle, but that primordial follicle contains a primary oocyte. And that primary oocyte has been arrested at the prophase stage. Until at the time of puberty, at prophase stage. So we have some inhibiting factors that are released to inhibit further development of the primary oocyte. And then thereafter, at puberty, you then have the development of the follicles as primary follicle. Here, the primordial follicle will be having flattened cells that we refer to as the follicular cells. Under the influence now at puberty of follicle stimulating hormone, we now have those flattened cells becoming the granulosa cells, forming the primary follicle, the secondary follicle, solid secondary follicle, and then vesicular type of secondary follicle, where we now have the androgen developing. And then finally, after ovulation, we now have the mature follicle, Graafian follicle. After ovulation, you then have the corpus luteum being formed under the influence of luteinizing hormone. However, for the oocyte, already it will have undergone meiosis, but it has been arrested at the prophase stage. It will only undergo the second meiotic stage after fertilization. So we have the first meiotic stage, meiosis. And after this first meiosis, this is what forms what we refer to as asymmetric form of meiosis, where we have a polar body. So we have the first polar body after the first meiotic stage. The first polar body does not contain a lot of cytoplasm. It's like a residual body. Then after that, after fertilization, that is when you now have the second meiotic stage, where you have the second polar body and now the ovum. So from one oogonia, you get only one ovum, unlike what we saw in the case of the testis, where from one spermatogonia, one spermatogonia B, you'll get 4 spermatozoa. But from one spermatogonia, you get millions and millions of spermatozoa. So this is the difference between what occurs in the males and in the females. Now we talked about corpus luteum or pregnancy. This is a corpus luteum. We saw the other day that the steroid secreting cells will normally have poorly staining, polygonal in shape, and the cytoplasm will appear vacuolated. So these are the lutein cells. Let's now talk about the oviduct. The oviduct is made up of 4 parts. We have the infundibulum, The infundibulum is a funnel-shaped part, and that funnel-shaped part comes to lie close to the ovary, and the infundibulum consists of finger-like projections that we refer to as the fimbria. So these finger-like projections are what we call the fimbria. One of the fimbria attaches to the ovary. So we have a fimbria attaching to the wall of the ovary. And then remember, the ovary is also attached to the posterior abdominal wall by suspensory ligament and the round ligament of the ovary. The suspensory ligament contains the ovarian artery, but the suspensory ligament also rotates the ovary so that the part which is ovulating is directly opposite this infundibulum, the funnel-shaped part. So the finger-like projections are to increase the surface area and to increase the suction because the cilia are beating towards the other inner part of the oviduct. And like I said, this part is open to the abdominal cavity because the Mullerian duct was actually opening to the abdominal cavity. The other part is the ambula, which is the largest part. The most dilated part is the ambula. And this is where fertilization takes place. The mucosa of the ambula is thicker and more folded mucosa. And then we have the isthmus, which is a narrow part. The isthmus of the uterus, of the oviduct, consists of a thicker circular muscle, and therefore acts like a sphincter. So within the isthmus, this acts like a sphincter. And then we have a very small part which traverses the uterine wall. This part that traverses the uterine wall we refer to as the intramural part. Now notice here we have the infundibulum funnel shape. Here is the suspensory ligament of the ovary, and that suspensory ligament rotates the ovary so that the ovulation part comes to the ovarian fossa. The ovarian fossa comes to lie completely next to this infundibulum. So what is the internal structure? What do we talk when we talk about the section through the oviduct? The oviduct consists of a mucosa, and the mucosa is lined. The mucosa is a highly folded mucosa and is lined by simple pseudostratified columnar epithelia. This pseudostratified columnar epithelia consists of 3 cell types. It consists of columnar ciliated cells, and of course the cilia is supposed to create a current by moving the fluid towards the uterus. It beats towards the uterus. The other cells are non-ciliated secretory cells. So the non-ciliated secretory cells do not have cilia, but they are secretory. So those have very well developed Golgi apparatus, rough endoplasmic reticulum, and some smooth endoplasmic reticulum. So they secrete fluid in which the ovary is going to be surrounded as it moves towards the ambula for fertilization, and also the zygote as it moves towards the uterus. The other cell types are the basal cells. Basal cells. The basal cells are undifferentiated cells. Undifferentiated cells. So this can differentiate to form the secretory and non-secretory cells. So they act as stem cells. Immediately below the epithelia, we have a lamina propria. Lamina propria, consisting, just like all lamina propria, of loose connective tissue and is a highly vascularized area. Loose connective tissue and a vascularized area. After that, we have a muscular layer, consisting of inner circular and outer longitudinal layer of smooth muscle. Smooth muscle. This smooth muscle is very important in movement of spermatozoa and movement of the fluid. This movement is by peristaltic activity. It is movement of the ovary, movement of the oocyte, movement of the zygote after fertilization, and also it is very helpful in orientation of the spermatozoa. As the fluid moves towards the uterus, the spermatozoa is moving against the movement of the fluid, it's countercurrent. That to some extent is a selective process in which the spermatozoa that are defective are not capable of swimming through to reach the ambular part of the oviduct. Ambular part of the oviduct. So the most fit spermatozoa are the ones which reach the ambular part of the spermatozoa, ambular part of the oviduct. Like I've said, the difference there is at the isthmus, we have a thickened circular muscle which acts like a sphincter. Then the intramural part is just that part that traverses the uterine wall. For the uterus, the functions of the uterus, you remember, we talk about formation of the placenta. And as a result of this formation of the placenta, it's very important in the nourishing of the developing fetus. It's also very important in physical protection of the developing fetus. The nourishment, physical protection of the developing fetus, and the muscular wall of the uterus is very important in the expulsion of fetus at term. Expulsion of fetus at term. The uterus also is very important in transport of spermatozoa, important transport of the spermatozoa. We remember we talked about release of prostaglandins during ejaculation, and this can cause mild contraction of the uterus, which will be important in the transport of spermatozoa through the uterine cavity all the way to the ambula of the oviduct, where fertilization takes place. I sometimes call this endometrial cycle a cleansing activity. During the menstrual cycle, there is desquamation of the functional layer of the uterine wall. That desquamation would remove any debris, possibly any bacteria, any substances that have been attached to the inner wall of the uterus. And then formation of the placenta. After implantation, we have the formation of the decidual cells. So the decidual cells are very important elements in hormone production. So the uterus, after implantation, is what we call decidua. The endometrium becomes a decidua. It transforms to become a decidua. So this wall of the uterus, we have several layers. we have what we call the endometrium. The innermost wall is the endometrium. And the endometrium is lined by an epithelia, which is columnar, sometimes ciliated epithelia, especially in the child, and contains endometrial glands. These are tubular glands that invaginate from the epithelia. Then we have a lamina propria. The lamina propria is just like what you've seen before, a highly vascularized region comprising of blood vessels, a lot of blood vessels, some connective tissue, and also the glands in the endometrium are within the lamina propria. During the endometrial cycle, we have the active stage where the endometrium is divided into two parts. The innermost part is the stratum functionalis, and that stratum functionalis is the part that is shed off. It is shed off during the menstrual cycle. All the glands in the stratum functionalis are shed off. The blood vessels in stratum functionalis are also removed. The glands at the basal part are the ones which now regenerate to form the stratum functionalis. At the peak of the endometrial cycle, during the secretory phase of the endometrium, the stratum functionalis can be divided into two parts. The innermost layer is what is called stratum compacta, very close to the lumen. These contain openings of the endometrial glands and also has more connective tissue. Opening of the endometrial glands and more connective tissue. We refer to this as stratum compacta. And below that is the stratum spongiosum, and stratum spongiosum contain the endometrial glands, with a lot of blood vessels, with less connective tissue compared to stratum compacta. Stratum basale, or the basal part of the endometrium, is what regenerates after menstruation. So this part is not shed off. Then we have the myometrium. Remember, endo means inside. Metra pertains to the uterus. Myometrium, myo means muscle. Metra pertains to the uterus, that is muscles of the uterus. This is made up of smooth muscle. So we have a myometrial layer made up of anastomosing type of smooth muscle. No very clear circular and not very clear circular or longitudinal muscle. Not very clear. Sometimes it might appear spiral. That is smooth muscle. Within this area, we can look at it in terms of the blood vessels. Some areas we have very small capillaries, then some areas we have very large blood vessels, and so on. So that can be reclassified further. And then we have a subserosa. We are using the word subserosa because the uterus is covered by a peritoneum, which is a serous membrane, a subserosa. And this is made up of dense connective tissue containing blood vessels and also some elastic fibers and collagen fibers. And then the outer part is what we refer to as the perimetrium. Remember, peri is the outer, metra pertaining to the uterus. is a perimetrium. How is the blood supplied to this particular organ? We have the endometrial uterine artery. The uterine artery forms the arcuate artery, and the arcuate artery, after the arch, we have what you call radial arteries. The radial artery will form two layers. They can form a coiled artery. Coiled artery within the stratum functionalis is a coiled artery. This coiled artery extends to the epithelia here and forms subepithelial capillaries. The basal part from the radial artery occurs just after the myometrium. The basal part here, stratum basale, is supplied by straight arteries. You will understand soon the significance of coiled arteries supplying the stratum functionalis. So stratum functionalis is supplied by coiled arteries. When the arteries coil up, they are called coiled arteries, they constrict the blood supply to the stratum functionalis. So they deny blood supply to the stratum functionalis. When that blood supply is denied, this part undergoes ischemia, then there is degeneration of the cells. After that degeneration, that's now the menstrual flow. So all that part of the stratum functionalis is shed off because there is no blood supply to it because of that coiled arteries. But the stratum basale is constantly supplied by blood, the straight arteries. So this can now regenerate to form the blood vessels and also the glands to form the stratum functionalis. The subepithelial blood vessels now form the venous, open into venous lacuna, and this open into endometrial veins. So this blood supply is very important. You need to understand this to be able to explain what happens during the endometrial cycle. So let's look at the endometrial cycle. The endometrial cycle is broken down into several stages. We have the actual shedding of blood. Shedding of blood, what you call menstruation. It's not just blood, it's blood with all the desquamated tissues. All the debris are shed off. So that is what we are calling now the menses. So during that menses, that is shedding off of desquamated cells, blood also that because of the bleeding in the coiled arteries that are being shed off and so on, that occurs from day 0 to day 5. Then from day 5 to day 4, from day 5 to 14, this is the proliferative stage. We have desquamated, we have removed stratum functionalis. So here we now have regeneration of the spiral arteries, the vasculogenesis. Regeneration of the spiral arteries from the vessels at the basal part, stratum basale, and also regeneration of the endometrial glands. Regeneration of the endometrial glands from the remnants of the endometrial glands at the basal part. This is under the influence of estradiol and follicle-stimulating hormone. So proliferative stage is a follicular stage. follicular stage. So this regeneration continues. The next stage is secretory stage. Secretory stage. So I note that day 8, remove this day 8. Secretory stage. Secretory stage initially will be under the influence of follicle-stimulating hormone and also luteinizing hormone. Remember, luteinizing hormone is very important in the production of progesterone. So the progesterone is very important in this stage of the progesterone is very important in this stage of the secretion. So this we also refer to as the luteal phase. Luteal phase. In the early phase of the secretory phase, the glands are tortuous. The cells have a lot of glycogen in them. And towards the end of the early stage of the secretory phase, the glycogen is removed, and therefore the basal parts of the glands appears vacuolated because the glycogen is shed off. Why do we have this glycogen? This is in preparation for production of glycogen, which is very important in the nourishment of in preparation for implantation. In the mid-secretory, again, the glycogen completely disappear. The glands now become serrated, leaf-like, and they are tortuous. Leaf-like and tortuous, they have lengthened, so there's a thickening of the endometrium, specifically the stratum functionalis. There's a lot of mucus. The glands contain a lot of mucus within their cytoplasm. In the late secretory, there's a very distinct basal zone and also the spongiosum zone. The glands, again, at this stage are very tortuous. So here the glands are swollen because there's a lot of mucus in the glands, a lot of glycogen in the glands, in the cells, and there's also thickening. So the cell is very thick, containing mucus and glycogen, and that's why these particular glands are tortuous. So at this stage, the endometrium looks like edematous, looks like fluid-filled areas. At this stage also, there's an increase in connective tissue, and this is promoted by prolactin and relaxin. So at this stage, we have a very clear zona compacta with very dense connective tissue and the opening of the glands. That's what you call now the late secretory. After this late secretory, because we said the secretory phase is in preparation for implantation. After the late secretory, if there is no fertilization, there will be no human chorionic gonadotropin hormone release. There will be a reduction in the luteinizing hormone, and therefore regression of the corpus luteum. As a result of this regression of the corpus luteum, there is reduction in the progesterone. Remember, progesterone is produced by lutein cells. Reduction in progesterone. Progesterone was very important in this development of the secretory phase. So as a result of this reduction in progesterone, we now end up with the coiling of the coiled arteries. Then there is denial of the blood supply to the superficial layer of the endometrium, that's stratum functionalis, and then we have the menses. we have the menses. So we now have shading off of the stratum functionalis because of reduced progesterone and also coiling of the vessels, the coiled arteries that supply the stratum functionalis. So this is what we have as the endometrial cycle, also referred to. These are changes in the endometrium, but you call it endometrial cycle. But menstrual cycle also will include what is happening within the ovary of our endometrial cycle. What's happening within the ovary? Within the ovary, we have development of the follicles. Many follicles, 15 follicles develop at any given time, but only one mature follicle, ovarian follicle, is ovulated. This is because of, you can call it the dominant follicle, suppress the other follicles from developing so that you have only one mature follicle developing. That dominant follicle could be because it is receiving more follicle stimulating hormone. It's receiving more follicle stimulating hormone compared to the other follicles, so that it becomes more dominant, and now ovulation takes place. The development from the primary follicle all the way to the mature follicle at ovulation takes about 64 to 74 days. 64 to 74 days. So there are several follicles at different stages of development within the ovary. This is the endometrium, and in this endometrium, this is the lumen of the endometrium. These are the opening of the glands. The epithelia here is simple columnar. These are the endometrial glands, which are coiled tubular glands. Coiled tubular glands. So this part of the endometrium is what we call the stratum basale, the basal part, close to the myometrium, and the whole of this part here is the stratum functionalis. So the whole of this part is shed off during menstruation. Now this area where we have close to the lumen, this is what we are calling stratum compacta. A lot of more connective tissue, dense connective tissue. These other areas here is the stratum spongiosum. And the stratum spongiosum contain a lot of these glands, the tortuous glands. From the uterus, you go to the cervix, and the cervix is just like the uterus. Remember, the uterine glands contain a lot of glycogen and a lot of mucus. Normally, that is produced, goes to the vaginal part. The glycogen is broken down by anaerobic fermentation to form an acidic environment within the vaginal part. The cervix, sometimes cervix means neck. The cervix has two parts. It has an infravaginal part and supravaginal part. The supravaginal part is sometimes called endocervix, and the endocervix is just like the uterus. It's lined by simple columnar epithelia, a lot of mucosal folds. We have cervical glands producing mucus and also glycogen, just like what we have in the endometrium of the uterus. We have the lower part of the cervix, and the lower part of the cervix is lined by stratified squamous non-keratinized epithelia. Stratified squamous non-keratinized epithelia. The stratified squamous non-keratinized epithelia does not contain the stratum corneum like what you have in the skin. The cells, the squamous cells, contain glycogen in their cytoplasm. They contain glycogen in the cytoplasm. Immediately after the epithelia, we have a lamina propria, and within the lamina propria, we would have some loose connective tissue and some lymphoid organs, lymphoid cells within the lamina propria. These are the glands, and this is the epithelia, the lamina propria. So we have what is similar to inner part of the endometrium, the mucosa, submucosa, that contains glands, cervical glands. We have a transition zone. And in that transition zone, this is where we have the cells changing from columnar to squamous, columnar to squamous. So the columnar cells of the inner part of the cervix, within the transition zone, we have a pseudostratified epithelia comprising of a mixed columnar cells, basal cells, and also some cuboidal-like cells, and then eventually transiting to form the squamous epithelia. So that is what we have in the cervix. I've said the endocervix is exactly like the uterus. The exocervix is the part that projects into the vaginal part. For the vagina, remember we had two parts. It was originating from two parts. The upper part was from the lower part of the Mullerian duct, the upper part, and the lower part is from the sinovaginal bulb, which is a thickened endodermal layer that grew towards the Mullerian duct. And that sinovaginal bulb is within the definitive urogenital sinus. Definitive urogenital sinus. It's lined by a mucosa, which is stratified, non-keratinized squamous. Stratified, non-keratinized squamous epithelia that contain, the epithelia contain glycogen in the cytoplasm. The lamina propria has dense connective tissue, in this particular case, with a lot of elastic fibers and some venous plexus. Elastic fibers and venous plexus. It's what we have in the lamina propria. Amongst those venous plexus and also the dense connective tissue, we have the wandering cells. These are cells of the immune system. The mast cells, the eosinophils, neutrophils, basophils, macrophages, all these are there to counteract any infection that could penetrate through into the inner part. Then we have a muscular coat comprising of inner circular and outer longitudinal layer. Inner circular and outer longitudinal layer, except at the opening. Except at the opening, that's close to the vestibule, which contains skeletal muscle or bulbocavernous muscle. That is close to the vestibule. And the outermost layer will be tunica adventitia, consisting of loose connective tissue. And this is where the blood vessels, lymph, and nerves enter at the tunica adventitia. Do you have any changes during the endometrial cycle? Under the influence of estrogen, the cells become thick and polygonal, with an increase in keratohyalin granules. And then at the time of ovulation, we have an increase in the glycogen, and desquamation. When we have progesterone, the epithelia becomes thin and desquamates. So this desquamation of the epithelia, that means sloughing off of the outer cells. And the cells in that case are basophilic because they have a lot of glycogen in them. So you can actually try to detect the endometrial cycle by taking swabs from the vaginal part, and you can also be able to look at these cells, and from looking at the morphology, you can say there's either an estrogen stimulation or progesterone stimulation. Also, at the transition zone in the cervix, I've said transition zone is where the endocervix transits to form the exocervix. This is where we have the cells changing from simple columnar to squamous, non-keratinized epithelia. So that transition zone, the cells can undergo metaplasia. And it is that region that we normally have development of cancer tissue. At the transition zone. So you can take up the cells in what they call swabs, and you can look at those swabs in what they call stain with Papa-Nicolau stain, and know whether the cells are cancerous or not cancerous. That's how they detect cervical cancer. For the placenta, after implantation, the placenta now changes, the uterus now changes to form deciduum. It's called deciduum. The placenta is formed by both membranes of the fetus, the chorion, and also formed by the maternal part. So it has fetal part and maternal part. What would be the functions of the placenta? One, exchange of gases, metabolites, and electrolytes. From the mother, remember, the mother is the one supplying all the nutrients, all the gases. Exchange of the fetal waste products. The fetal waste products are absorbed into maternal circulation and transported to the maternal kidney, where they are removed. And then production of placental hormones. So human chorionic gonadotropin hormone from the trophoblast cells, somatotropin hormone, human chorionic thyrotropin hormone, the estrogens, the progesterones, all these are produced in the placenta. So that in the third trimester, you can remove the corpus luteum and pregnancy will still be carried to term. Of importance is also transmission of maternal antibodies. So maternal antibodies are transmitted to the fetus. So that during pregnancy, sometimes mothers are vaccinated, and that vaccination is to ensure that the fetus has adequate protection at birth. And then secretion of fibrinoids. Now, fibrinoids is fibrous, homogeneous tissue-like secretions that accumulate at the junction between the fetal and the maternal part of the placenta. So they increase. And this is the area where there's separation of the placenta after delivery. So the placenta consists of the chorionic villi. Remember, I think I've done that in embryology, where we have, after implantation, we have the trophoblast penetrating into the endometrium. And where you just have the trophoblast alone, we call that the primary chorionic villi. Then you have the trophoblasts forming both syncytial trophoblast and the cytotrophoblast. And then the mesenchyme tissue, the intermediate intraembryonic mesoderm that now penetrates into the primary chorionic villi to form the secondary chorionic villi. So there's differentiation of that mesenchyme tissue and the blood vessels that now form the tertiary chorionic villi. So this chorionic villi here will be having one blood vessels from the maternal part, fetal part, fetal blood vessels. It will be having mesenchyme cells, and is surrounded by the trophoblasts. So these trophoblasts will be syncytial trophoblast and cytotrophoblast. Some books talk of in the third trimester, you have only a single layer of trophoblast, but that has been distributed. Currently, the theory is that all the two layers are there, both cytotrophoblast and syncytial trophoblast. And these chorionic villi are surrounded by maternal blood. So this maternal blood, so there's no mix-up between the chorionic, between maternal and fetal blood. Maternal and fetal blood is separated by the trophoblast cells. This just to illustrate what I'm talking about. This is fetal blood from the umbilical artery, umbilical vein. So the umbilical vein picks up the blood from here. It will be having the oxygenated blood. It will be having blood with a lot of nutrients. So these are the vessels. Now you need to note that as the chorionic villi become tertiary, then on mature chorionic villi, then we have a very thin separation between the maternal and fetal blood. So this constitutes what you call blood placental barrier. So this is a barrier from maternal blood between the placenta and the maternal blood. So around this clear area, this is where we have maternal blood. So there's no mixing completely. What happens to the endometrium after implantation? The decidio basalis, the decidio parietalis, the endometrium now transformed. The part that we have the implantation, we refer to as the deciduum basalis. The rest of the endometrium, we refer to as the deciduum parietalis. And then the endometrium, immediately after the implantation, when the blastocyst is completely embedded, then that endometrium that surrounds it is what we refer to as the deciduum capsularis. But as the pregnancy continue, deciduum capsularis and deciduum parietalis fuse together. But if you take a section through the uterus, the deciduum comprised of the compactor zone, zona compacta, containing of decidual and trophoblast cells. Zona spongiosum, what was zona spongiosum? Consists of flattened endometrial glands opening into the surface, and then zona basalis, what was initially the stratum basalis, consists of narrow glands near the endometrium. So zona compacta and zona spongiosum constitutes what we call the basal plate. The basal plate. And then this is what forms the deciduo-trophoblastic complex. For the breast is an accessory gland, and the breast is a modified sweat gland. Modified sweat gland developing by invagination of the ectoderm. So there's that invagination of the ectoderm, and then continues on invaginating into the mesoderm. So there's invagination of the ectoderm, and that invagination, the initial part of the invagination, will form the lactiferous duct. Lactiferous duct. Then the cells around that area will grow and form the nipple. As a result of growth, they form the nipple. And then we have the lactiferous sinus, and then the aqueous-secretory part will be the alveolar. The alveolar. The alveolar will have intralobular ducts and then interlobular ducts, which open into the lactiferous duct, and the lactiferous duct open into the lactiferous sinus at the areola. What are the phases of development of the mammary gland? There's before puberty, during puberty, and the adult stage. Remember, during puberty, there's proliferation under the prolactin and also under the influence of progesterone. There's proliferation of the mammary gland, so that there's an enlargement of the mammary gland. And then in the adults, we have several phases. There is the resting stage, the pregnancy stage, and lactation during the production of milk. And then all of these glands undergo involution, so that during the senile stage, after menopause, where the influence of the hormones is lacking, we have involution, and a lot of it is replaced by adipose tissue. This is the mammary gland with the septa. These are the alveoli. This is the duct, intralobular duct, and the alveoli is lined by simple columnar epithelia. Simple columnar epithelia. Now, the actual secretion of the mammary gland is in two. We have the merocrine type of secretion, and we have the apocrine type of secretion. Merocrine type of secretion and apocrine type of secretion. In apocrine type of secretion, this is where the apical part of the cell, together with the secretory product, is released. And this is common with the lipid. And then for the proteins, this is by merocrine type of secretion. So remember, the mammary gland produces proteins. It also produces lipids. And therefore, the organelles are mixed organelles that produce proteins, lipids, and also some carbohydrate. The gross anatomy people address for you the actual lymphatic drainage is very important in cancer. So the regulation of the mammary gland in terms of development, we have the mammogenesis during the development of the mammary gland, where estrogen plays a major part in the development of the duct. Steroid hormones, growth hormones also play a major part. And then the lobules, we have progesterone as the main driver for the lobulation, lobules and alveolar. Progesterone are the main. That's why progesterone, as it prepares for pregnancy and maintains a pregnancy, also prepares the mammary gland for feeding the child. Estrogen to some extent, but progesterone is the main driver in the development of lobules and alveoli. Then the galactopoiesis, the actual maintenance of production of milk, that is by suckling. So suckling is very important, and also prolactin plays an important role in terms of production of milk, galactopoiesis. And then here, galactokinesis is where you are ejecting the milk. And during that ejection, remember we talked about the role of oxytocin in ejection of milk. So we have supraoptic nucleus and paraventricular nucleus producing oxytocin. And that oxytocin acts on the myoepithelial cells that are situated between the alveolar cells and the basement membrane of the mammary gland. So those basket cells or the myoepithelial cells contract and eject milk. That is because of oxytocin. It's not enough tissue situation, but it is oxytocin. But remember, the hypothalamus receives information, sensory information. So by suckling, you are stimulating sensory information from the hypothalamus and from the breast is sent to the hypothalamus, and then the hypothalamus produce more milk, and in the posterior part of the pituitary, the milk is released. And then maintenance of the mammary gland. Maintenance of the mammary gland is under the influence of prolactin. Prolactin is the main driver, but all these other hormones play a role in a fully developed mammary gland. So you can see that hormones, prolactin features in virtually a lot of development of the mammary gland, right from the development of the ducts, maintenance of pregnancy, maintenance of lactation throughout the breast functioning. Fine, I'll invite any questions. Should anybody have any questions, please do ask. I have a question in the chat about paramesonephric duct. Paramesonephric duct develops lateral to mesonephric duct. Lateral to the mesonephros, that's mesonephric duct. It develops as invagination of the coelomic epithelia. So coelomic epithelia cells invaginate into the mesonephros. As it invaginates as a solid core of cells. That solid core of cells invaginating eventually canalizes. That means it now attains a lumen. It attains a lumen. And that lumen is now the lumen of the oviduct, the uterus, and the upper part of the vagina. So that is about the development of the paramesonephric duct. It is called paramesonephric duct because it develops lateral to the mesonephric duct, adjacent to mesonephric duct. And the mesonephric duct, like we had said, develops as an epithelial thickening of the coelomic epithelia. Corpus cavernosum, there are two bodies in the corpus cavernosum. If you take a section through the penis, there's a corpus cavernosum. These are two bodies comprising of erectile tissue and cavernous spaces. Corpus spongiosum is the part that surrounds the urethra, the spongy urethra. The part that surrounds the spongy urethra. That's what we refer to as corpus spongiosum. Now, corpus cavernous, the male, the testis. However, in the case of the female, sometimes they talk of by default